All right, so I'll start now. So hello, everyone. My name is Rechu. I'm doing my PhD at UK with Professor Tim Carroll and Professor Guy Wallace. And today I will be presenting the main results from one of our experiments. As the title suggests, we found that the early corticospinal excitability changes during visually guided reaching reflect the stimulus direction rather than the reach direction. And in this brief presentation, I'll try to tell you how we arrived at this conclusion. Now, before I delve into the experiment, I wanted to introduce the reaching task that we used. In this task, participants sat in front of a monitor with an air sled supporting their forearm. A pulley exerted a 5 Newton load force to the right on the arm, which helped to increase the baseline activity of the right pectoralis major muscle. And we used surface electrodes to record EMG from the other 3 muscles listed here. And on screen, this is what participants saw. The task was to reach towards or away from the black target when it briefly flashes beneath the barrier on either side of the fixation cross. So in this case, the reach is made towards the target, and we call this a pro-reach trial. And so in a task like this, if you look at the activity of the muscle, if you look at the activity of muscle in the pectoralis major muscle during pro-reaches, so here, the bottom row shows a raster plot in which the EMG amplitude is colorized, with yellow being high activity and blue being low activity. And the trials are aligned to the onset of the visual stimulus, which is represented by the red line. And the white circles you see on the raster plot are mechanical reaction time of the arm measured using an accelerometer. And the trials were sorted based on reaction time, with the fastest trials being at the top and the slowest ones being at the bottom. And the top panels here show the average EMG activity from all the trials at the bottom. Now, as expected, around the mechanical reaction time value, there is consistent activation of the pec for left reaches and an inhibition for the right reaches. However, in both left and right reaches, you notice some time-locked activation or inhibition, which starts around 80 milliseconds and peaks around 100 milliseconds. That seems to be separated from the subsequent voluntary EMG burst. So this short latency response seems to be tuned towards the location of the visual target, and it occurs consistently at around 80 to 100 milliseconds, irrespective of the manual reach. Now, what happens if we ask the participant to move away from the target or execute what we call an anti-reach, as shown here? Now, when the target is on the left side, the person has to reach away to the right. And if you look at the pattern of EMG in that case, we see an appropriate pattern of muscle activity around the onset of the hand movement. So for the left target, the pec is inhibited, while for the right target, the pec is activated. However, we also see that the early time-locked activity that we saw in the pro-reach condition is still preserved. And most importantly, the early activity is in the opposite direction to the later voluntary activation that we see. So what we see here is that in both pro and anti-reach condition, the time-locked, there appears to be a time-locked activity that is tuned towards the direction of the visual target. And this response has been termed as a short latency response, or SLR. And over the last decade or so, several studies have attempted to understand its neural mechanisms. Now, the obvious question is, where is the signal that causes this response coming from? The first possibility that comes to mind is a fast cortical pathway that goes to the motor cortex. However, we and other groups have speculated that a subcortical pathway involving the midbrain superior colliculus and the brainstem reticular formation might underlie the short latency response. So my experiment addresses the origins of the short latency response. So the aim of this experiment was to test if corticospinal excitability around the short latency response time window is consistent with the spatial properties of the SLR, which would mean that it changes as a function of the target location as opposed to the reach direction. So we adopted the moving target paradigm and delivered TMS pulses at latencies around the window we see this short latency responses, as participants performed either pro or anti-reaches. On separate trials, TMS pulses were delivered at one of 5 latencies around the SLR time window, or at a latency 30 milliseconds before the estimated onset of the voluntary EMG. And we also had trials in which no TMS was delivered, so as to measure the activity of the muscle in the absence of TMS. Now, if you look at the results, for the pro reaches, we saw that from 70 milliseconds onwards, the MEPs were enhanced when the pec muscle was the agonist and inhibited when the pec was the antagonist. A Bayesian ANOVA confirmed this by providing strong evidence to indicate that the MEP amplitudes depended on the interaction between stimulation latencies and target location. So here is a zoomed in version of that image showing the results of the ANOVA. So if we compare the MEPs with the background EMG in the absence of TMS, we see that there is a little bit of separation between the EMG trace for left and right targets. However, ANOVA results showed inconclusive evidence to say that background EMG changed consistently as time progressed. However, because there is a subtle difference towards target-related modulation in background EMG, it remains possible that there is some contribution of the background EMG change to the dissociation of the MEP that we observe. Now, now things get interesting for the anti-reach. So at 70 and 80 milliseconds, we see that the MEP is biased towards the stimulus direction and not to the direction of the reach goal. However, by 90 milliseconds, this pattern tends to flip, and by 100 milliseconds, which is just prior to the voluntary reaction, the MEP reflects a goal-directed response. And if you zoom in, this pattern is much clearer. And if we compare the MEP to the background EMG, we see that there are no significant difference until about 100 milliseconds. And there is a trend to flip in activity that is broadly related to the MEP results that we saw. So while the background EMG does not exactly have the same pattern as the MEP results, there are some changes that could be related to the MEPs. Now, if you look at results from other muscles, for example, posterior deltoid, which is the antagonist of pec muscle in this task, it shows an almost exact opposite trend to the pec results. While if you look at functionally irrelevant muscle like the FTI, we see that there is no visual stimulus-induced bias in excitability. So to summarize, we see that during visually guided fast reaching, corticospinal excitability responses at 70 and 80 milliseconds after stimulus onset reflects the direction of the visual stimulus, irrespective of the task goal. And corticospinal excitabilities from 100 milliseconds reflect the motor goal, irrespective of the physical location of the visual stimulus. And finally, the target responses seem to occur only in contextually relevant muscles. Now, we saw that the target appearance influences the corticospinal tract as early as 70 milliseconds after the target onset. However, it is still unclear where this modulation occurs. It could occur at a cortical level, or it could occur somewhere at the subcortical level. So in our next experiment, we intend to address this question by comparing modulation of responses to transcranial electrical stimulation and transcranial magnetic stimulation. And this should provide evidence about the contributions of cortical and subcortical structures in generating these extremely short latency visuomotor responses. That is all. Thanks, everyone. Any questions?
Thanks, Rechu. Well done. We do have a question from Kate Hoy. So she's asked, Rechu, great talk. What do you think is responsible for the background change in EMG in the absence of the TMS?
That is a really good question. I mean, so the task that we have designed, the emerging target paradigm, is shown to promote these fast, short latency responses. So the background EMG that we could be seeing is probably because of the these visual, like the stimulus-induced responses generating some sort of EMG response. Yeah, that would be my guess.
Okay, thank you. I guess I was also going to ask about the EMG. So you did see some subtle changes in EMG. Did you have any opportunities to follow that up to see how likely that, whether, could the EMG be underlying all of the main biases in TMS that you saw, or is that unlikely?
So we were concerned about this. So what we did was we we took all the subjects and split them based on the level of activity that we observed in the early latencies. So we had a group of participants who showed high activity and then a group of participants who had low activity. And we could still see the pattern of MEP results even in the participants who had like relatively flat EMG, background EMG in the early latencies. So we are pretty confident that the EMG is not driving the pattern of MEP results that we are observing.
Okay,